Kim, and thank you, Anne, for handling all of these things. So it's very exciting to be in Function Cozy. This is the work of Rashi Karamola from my group in collaboration with Ido Friedberg's team. As some of you know, our group, my group and Ido's group have been involved in protein function prediction and a lot of technical work over the years. But this is an unusual work for us, in which case we tried to take a look at this field in general and try to understand how it functions as a field. And therefore, we conducted a survey of this field. So as a part of this research, we identified that there are 3 communities of scientists involved or related to protein function prediction. And one is a group of experimental scientists, biocurators, and computational biologists. Experimental scientists, as you know, they carry out wet lab research, they test hypotheses, they publish the results of their work in papers. Biocurators start from these papers, they systematize, process, organize this information, they put it in knowledge bases, they also develop ontologies, and so that this information is really presented in a good form. For perhaps downstream users, computational biologists, we start generally from this curated information from biocurators, and then we develop tools for the analysis and prediction of protein function. And then maybe we can use these tools by, let's say, experimentalists to guide further developments or further studies of protein function. So objective of our work was to conduct survey to assess state of affairs of this field. We wanted to understand their views of this community to the field, the practices, how they conduct their research. We wanted to assess the quality of protein function prediction methods. I will say a little bit more about that, especially the usefulness to experimental scientists. We want to understand how much these scientists in these communities collaborate in this field and how they work together. And then throughout all of that, we wanted to highlight positives and, of course, reveal the bottlenecks in the field. So we did the survey, and our rationale was, okay, so experts can give us the best information how we can understand this field. So our inclusion criteria are PhD in the field and publications in the field. So we exclusively interviewed PhDs. So as you can see here, we contacted 169 scientists, 55 experimental biologists from 25 institutions, of which we had 22 participants from 12 institutions. And so in total, if you look at biocurators, we have 19 participants from 10 institutions, and from computational biologists, 41 participants from 38 institutions. So some of you, maybe all of you, have been contacted, and we thank you for your participation. If you have not responded, that's also okay. We had 82 participants in total. So our survey was a Qualtrics form. It was done online. We offered each one of you a $10 gift card, and we hope that you use them if you completed the survey. So for how did our surveys look like? So for experimental scientists, we had a general questionnaire. We also had general questionnaire for biocurators and another slightly different general questionnaire for computational biologists. Why I call it general? Because everybody in the same group, biocurators, computational biologists, experimental scientists, had the same general questionnaire, but among these groups, those are slightly different. Now, for experimental scientists, we also had two personalized questionnaires. One we called specific questionnaire, where we gave them predictions of modern tools for protein function predictions and asked them to evaluate every prediction. Is this a useful prediction for them? Do they know already this is the true function for this protein, etc.? And then we also, for experimental scientists, had another personalized questionnaire called comparative questionnaire, where we side by side gave them predictions from the state-of-the-art tool and a baseline tool, and we asked them to tell us, can they see the difference between these two? It's not so obvious. So here's a little bit about our cohort. So this slide shows participant experience in training. As you can see on the left-hand side, this is a fairly experienced cohort. Most of our participants have more than 10 years of experience in this field. And the blue bars are always going to be experimental scientists, the red bar are going to be biocurators, and the yellow bars are going to be computational biologists. If you see their fields of training, so this is general information we solicited, you will see that most experimentalists and biocurators have been trained in biology. Most computational biologists have been trained in computer science, although some computational biologists have training in biology and some biocurators have training in computer science, just as you would think that it would be. So we asked them some general questions. We showed them a number of databases that are related to protein function prediction or protein function annotation in general, and we asked them to rate their familiarity with these databases. So 4 would be used frequently, this database, and 0 would be they're not familiar with it. By and large, and you can see later these results in the paper, These biocurators and computational biologists are most familiar with all of these databases, a little bit more by biocurators than by computational biologists, and experimental scientists are generally less familiar with almost all the databases except Protein Data Bank, and you will see it as the fourth one from the left. Okay, experimental biologists use Protein Data Bank quite a bit. With regarding to the software, some general tools in the field, we see no difference between these three communities and their familiarity with BLAST. So the scores are here on the right-hand side at the bottom, and you will see that we do not see any difference. So everybody's familiar with BLAST, and we see similar results for clastal and tools like that. So general tools, everybody knows about them. Now our main questions. We wanted to ask experimental scientists, do they use any software to understand function of a protein? First of all, 64% of them do not use any software. And of those 36% who do use some software, we asked them what software do they use, thinking that they would tell us some advanced tools for protein function prediction, and 0 of those were listed. So what they use is SciBlast, HHPred, String, SwissProt, etc., iTaster. So some of these are protein structure predictors, others are databases, and of course, Interpro and SwissProt can be, do have some predictions. So these experimental scientists can see predictions, but by and large, they do not see any of the advanced tools that we develop in this group. I'll come back to that point later. Now, we gave them side-by-side predictions. For each experimentalist, we looked up the protein they study, and we used one of the state-of-the-art tools listed here and selected randomly, and gave them top 25 predictions in molecular function, biological process, and cellular component for 4 different proteins, 2 of them that they understand really well, and 2 kind of general proteins. And we asked them to label each prediction as known, useful to them, surprising and possible, surprising and doubtful, wrong, or they can leave it empty, and that we call unlabeled. Okay, so, well, I did upload the video. Ido helped me upload the video, so the video is online. It's 14 minutes. It's a little longer, but I hope it's going to be okay. So we are now continuing with the specific questionnaire for biologists. Then we asked them a very simple question. Did the algorithm do a good job? I had that before. In my Zoom class, students muted me at one instance. So I'm quite used to that, so no problem. Yes, the reality of the field, there's optimism, but there's concerns. So Gaurav, let me, well, I don't know how to shift the slides now. Yes, you either get audio or visual, so not both. I'm sorry. I'll try to touch only one button here. Yeah, okay, so now we show them side-by-side predictions from the state-of-the-art tools and the tools that we labeled here as baseline. And you see these 3 tools, we again selected randomly one of the baselines according to CAFA. We know from CAFA experiments that state-of-the-art tools are better based on the metrics that we select. However, what we see here is that the experimental biologists are about equally ranking the wins and losses between the state-of-the-art and baseline tools. They are unable to see the difference between these tools. You might think of this as a concern. However, we only show them the top 25 terms, and so it is possible that the terms that actually distinguish the performance between state-of-the-art and baseline tools are actually below the 25th term. By and large, there is no difference. So now I move to the ontologies. Now we asked all 3 communities 2 different questions. The first question was, how useful do you think is a Go annotation for a protein, for an experimental scientist? As you can see, we gave them 4 possible answers: not useful, somewhat useful, moderately useful, and very useful. Well, experimentalists generally think it's somewhat useful to them. Biocurators are very enthusiastic. They think it's very, very useful. They think ontologies are very useful for experimental scientists, and computational biologists are more similar to biocurators, but kind of towards experimental biologists. And then we asked them a question, how well do you think Go terms describe protein function? We again see biocurators optimistic. They say very well, most of them, and then 25% or so say well enough, whereas computational biologists and experimental scientists are sort of centered around well enough. So they're not thinking ontologies are bad. However, we see a little bit more optimism amongst biocurators. Now here we interviewed mostly, in this case, biocurators and computational biologists, and we asked them 2 different questions. One was, what are the chief bottlenecks of the field? And on the left-hand side, you will see we gave them options: available data, ontologies, methodology, evaluation, and other. When we look at available data and ontologies, we see that more computational biologists think that this is the chief bottleneck in the field than biocurators. So computational biologists blame biocurators. Biocurators think that methodology and evaluation are the chief bottlenecks in the field. And about equal number of them say it's something else. And then we asked them, what is that something else? Very useful answers. One was explainability of the field. Another one was awareness of the field. One person said, converting protein identifiers between different databases is the chief bottleneck of the field. I thought that was really great. Yes. We suffer on that a lot. And so on the right-hand side, we asked them how much they collaborate with experimental scientists, and we see that biocurators actually collaborate the most. There's no statistical difference between these distributions, but they roughly collaborate more with experimental biologists than computational biologists do. And so I will try to briefly summarize this work. We surveyed the field. There is much more in the paper, which is accepted in Bioinformatics Advances, but is available on bioRxiv here on the right-hand side at the bottom. We identify many common views between these communities, but also some significant differences. We see that a lot of challenges in this field. For example, experimental biologists do not use the modern tools for protein function prediction, but if they were to use those tools, they would actually benefit by guiding their future research. They see many of these predictions to be surprising and possible or useful. On the other hand, biocurators develop ontologies that are not as much appreciated by the other two communities. And at the same time, it is very difficult to develop protein function prediction without standardizing this. Same for computational biologists. We do not produce tools and advertise them enough for biocurators and for experimental scientists to actually benefit from them. So social challenges in the field perhaps stand in the way of technical progress in the field. So I will just stop here with the limitations. You can see them on the screen. Thank you very much for listening to me. Apologies for the glitches. Thanks to Rashika and Ido Friedberg and NSF that funded this work. Thank you.